So we are able to hear because we have the organ of cortex. The trick is to move the hair cells right here. In order to do that, we need to move the basilar membrane. When we move the basilar membrane, the hair cells are going to move because the tectorial membrane is basically fixed. So how do we move the basilar membrane? By moving the fluid that we have inside this cochlea. Okay, so if we scroll down, so we can see right here the hair cells, tectorial membrane, having there a nice figure showing you the hair cells, and physiology of hearing. So how are we able to hear? We can do that by looking at some figure. For example, we produce sound waves with our vocal cords. The sound waves go through the air and they go inside our auditory canal. They bounce against the tympanic membrane. The tympanic membrane vibrates. That vibration is transmitted to the malleus, incus, and stapes, and the stapes is connected to the oval window that is going to transfer that vibration inside your cochlea right here. Now, in the cochlea, the vibration of the three little bones is transferred inside into the fluid in the form of waves. The waves are going to go through the perilymph right here, and that perilymph is going to move the vestibular membrane. The vestibular membrane moves the endolymph right here in the cochlear duct. The cochlear duct goes and moves the basilar membrane in the scala tympani. By moving the basilar membrane, as we said, we are going to move the hairs. The hairs open channels for potassium, and that initiates a depolarization that sends the information to your brain using this nerve, which is going to be the vestibular cochlear nerve. So going back to the physiology of hearing, or how do we hear it, because we said the middle ears, we have the three little bones, transfer the information inside your cochlea, and inside the cochlea we have the cochlear cells that need to be stimulated, and we already explained that. Right here is the same information, outer ear sound waves, vibration here, and then right here is waves in the fluid, and that moves the basilar membrane that eventually transfers the information to the hair cells, and from the hair cells into your brain. So we scroll down, right here they are showing you how the hair cells move open channels for potassium to initiate the depolarization. Scrolling down, sensory coding, but that is what helps us identify if something is very loud or is not that loud, right? I mean, soft sounds and loud sounds, for example, because the tectorial membrane is stimulated in different areas. Cochlear tuning is the ability of our ears to allow us to hear some sounds better than others, how the information goes up to your brain. In here you can see that we said everything for hearing is in the cochlea. The cochlea uses the vestibular cochlear nerve to send information to the brainstem, from the brainstem to the thalamus, and from the thalamus into the primary cortex auditory area. Okay, let's keep scrolling down. Our next topic is equilibrium. There are two main types of equilibrium, static equilibrium and dynamic equilibrium. So we scroll down. We have this part right here, which is called the vestibule, and this part right here, which are called the semicircular canals. The vestibule is for static equilibrium. Semicircular canals are for dynamic equilibrium. In the vestibule, we're going to have two structures. These structures are going to be called the macula. If you look at this figure right here, what we need to do is we need to move the hair cells. In static equilibrium, we can talk about something called linear acceleration, which is the movement, for example, when you're inside your car. So you're inside the car, you don't move, but the car moves. Basically, it's a movement from left to right. How do you know that the car is moving? Because your utricle will help you realize that even though you are not moving, but the car is moving, and that you're going to be able to identify that due to the fact that we have that utricle. Now, what happens when you are inside an elevator? Same concept. You don't move, but the elevator moves. How do you know that you're moving? Because, in this case, the saccule, which is vertical, will help you with that. Again, when you move either with the saccule or with the utricle, either up and down or left and right, in that case, the movement will move your utricle or your saccule, and then this gel right here will lag behind, and because it lags behind, it will move the hairs. And when it moves the hairs, it initiates the transfer of information to your brain. So if you just scroll down, we're going to look now at the semicircular canals. In this case, semicircular canals are not for linear, left and right or up and down. In this case, when you are turning, like for example, when you are sitting down on a chair and that chair starts turning around, then that's when you use the semicircular canals. In this case, the concept is the same. You have a gel, in this case it's called crista ampullaris, and inside the gel you have a hair cells. So then what happens is when you turn around, depending on the plane that you're moving, right? For example, this person is turning around, your body is going to help you identify where you are turning or how you are turning, because we have these three different canals in different positions that will help us identify in what direction you are turning. What happens is that when you turn, the fluid inside this tube is going to push the crystal pilaris, as you can see right here, and then this pushing is going to be able to move the hairs. And when the hairs move, the hair cells here capture the information and they send the information to your brain using this nerve, which again is going to be the vestibulocochlear nerve. So if we scroll down, we can see there how the information goes up to your brain. And we said it right here that the information will go using your vestibulocochlear nerve to your brainstem, from there to the thalamus, and from there into the vestibular cortex that is going to help us maintain equilibrium. Don't forget that we have 3 different areas to maintain equilibrium. One of them is the eyes, the other one is the cerebellum, and the last one is going to be the ears. And that's about it. Okay, the next topic in this chapter is going to be vision. We're able to see light. Light, vision is the perception of objects in the environment by means of the light that they are going to emit or reflect. If there is no light, we cannot see. So right here you have the eyes, and in here you can see that the white part is the sclera. This part right here with the color of your eyes is going to be the iris, and this area right here is going to be your pupil, the black part in the middle. So if we keep scrolling down, we can see that the iris and the pupil are covered by this crystal and translucent area called the cornea. Also right here they are showing you that we have the lacrimal glands. The lacrimal glands produce a lot of tears in order to clean your eyes. It also has substances such as immunoglobulin A in order to defend us. Right, they produce the tears, and the tears come out of the eyes through the lacrimal sac into the nose through the nasolacrimal duct right here. We can see that in addition to those structures, we are going to have muscles associated with the eyes. So if these are the eyes right here, we have muscle on top. This is going to be called rectus. So this is superior rectus down here, inferior rectus here, medial rectus, lateral rectus. We also have an oblique on the top, means superior oblique, and another oblique at the bottom, inferior oblique. This muscle moves due to the fact that they are associated with nerves. In the case of superior oblique, it's the trochlear nerve. Lateral rectus is the optic nerves, and all the others are going to be moved with the oculomotor nerve. Don't confuse this nerve that moves the eyes with the optic nerve that is going to give us the ability to see. If we scroll down, we can see the anatomy of the eyes after the eyes have been cut, so you can see the inside. So right here you can see there is a space called the anterior chamber between your cornea and the lens, which sits right behind the pupil. Okay, behind the lens, you have this other space where you're going to have the vitreous humor. Okay, right here in the front, you have aqueous humor. Right here, you have the vitreous humor. If you look at this part right here, this is the wall of your eyes. Okay, it has 3 layers. You can see here external, middle, and inner layer. Okay, they are called tunics as well in some books. You're going to see them as tunics. This external area is going to be the sclera. The middle area is going to have a lot of blood vessels, as you can see here. Okay, it's a very vascular area. This middle part is also called the uvea. And well, this middle layer, it has 3 main parts. The choroid, which is this part in brown, where you have a lot of blood vessels. If you can see here, it continues with the ciliary body, which is going to be the second part. And the third part is going to be the iris. The ciliary body produces this muscular mass that is going to be able to hold the lens in place using the suspensory ligaments. So what's the job of the lens? The lens sits behind the pupil and it allows to adjust the light before it reaches the back of your eyes. The inner layer, right here in yellow, is what we're going to call the retina. The retina is where we have the receptors that will allow us to see. So if we scroll down, we said in there the tunics, we said we have 3. We have the outer or fibrous layer, vascular layer in the middle, and in the inner is the tunica interna, which is where the retina is. Okay, so optical components, we said we have aqueous humor right here in the anterior chamber, and right here we're going to have the vitreous humor. In the anterior chamber, we have aqueous humor. So if we keep scrolling down, you can see in there the lens, neural components. You can see here, this is your retina. In your retina, this is the area where the nerve is, and this area right here is the area that will allow us to see detail. So where the nerve is located is called the optic disc. This area is orange, and this will allow us to see. everything. This is what we consider the peripheral vision. This area where all these receptors are going to concentrate, they're going to be the macula lutea. The very dark area right here is going to be the fovea centralis that is going to give us the detailed vision, right? Or the ability, for example, to read. So this is peripheral vision. This is visual acuity. So we scroll down, we have in there the figure showing us the fovea centralis, the optic disc. So we keep scrolling down. Formation of an image obviously is going to be on the back of your eyes. Right here we can use this figure. So the light goes inside your eyes, go through your cornea, through your lens, vitreous humor, and goes to the back of your eyes where your retina is. Depending on how far or how close the figure or the image is going to be, your lens is going to adjust and making sure that the right image is going to reach the retina so you are able to see. So we use this figure to show you how the image is forming here, but this is for refraction. You don't need to know anything about refraction. Near response, you don't need to know that. You keep scrolling down. Now, this is the retina. What you have in the retina? The receptors. What are the receptors? The rods and the cones. Okay, so these are the receptors that you have in your retina. In addition to that, there are other cells such as ganglion cells and bipolar cells. So if we keep scrolling down, we can see here figures of these receptors. One receptor is the cones, and the other one is the rods. Cones is for a lot of light. Rods are for a little bit of light. Cones are, for example, during a sunny day, and rods are, for example, for spaces where there's not a lot of light, dim light. Remember that if there is no light, we cannot see. We also have to remember that some people can read, for example, but they cannot identify colors. If you scroll down, we can see there is a test called Ishihara's test for that purpose, which is this, and that is it.